My name is Wright, as has been introduced, and I work for the Institute of Medical Biology. And as you can see, I am a scientist. Now, the stereotypical view of a scientist is somebody who's very analytical, logical, maybe wild hair, not so much in my case, that has a penchant for wearing socks with sandals and is working away all day, every day in the laboratory. It's not generally associated with having a need for a creative flair. And with today's talk, I hope to try and dispel that myth for you and give you examples of how creative scientists are really shaping the world in which we live. Take that opportunity to take I very rarely wear a lab coat, I promise you. Okay, so to start with, I'd like to give you some very key definitions that I'm going to use throughout this talk. And the first is, what is science? Science is the intellectual and practical activity encompassing the systematic study of the structure and behavior of the physical and natural world through observation and experiment. Importantly, it builds upon the existing knowledge that we have as the human race and furthers it through the activity of science. And this was very eloquently put by Sir Isaac Newton in 1676 when he said, "If I have seen further, then it's by standing on the shoulders of giants." So how do we do it? We approach science using something called the scientific method. The scientific method, as a base, means that we have to observe first. We need to understand the current level of knowledge. From that, we need to think of new directions or new questions, formulate a hypothesis, a question or an idea that we can test. With experimentation, empirical experimentation, and that often requires several rounds of iteration and refinement to improve our hypothesis. Once we've collected a sufficient body of work or experimental results, we can then develop conclusions or solid theories out of that. Now, I'm aware that this probably sounds a little bit dry, and maybe I should consider putting my lab coat back on and stepping back into the stereotype. So let's put it slightly more exciting. I'll give you two quotes. One is by Dudley Herschel, who's a professor at Harvard University and a Nobel laureate, and he said, "You're exploring a question that we don't have answers to. That's the challenge. That's the adventure in it," which is a very nice way to put it. And also, Deborah Smith of Penn State University, when describing the scientific method, says, "In the quest to make sense of the natural world, scientists think of new ways to figure out, to approach problems, to figure out how to collect meaningful data and explore what these data could mean. New ways, figure out, explore, all of these have an element of creativity about them." So now I've defined science and how we do it. What about creativity? So creativity is the use of imagination and/or original ideas to create something new and valuable. And if we take examples from the art world, it's very easy to see where creativity sits. We start with a blank canvas and do a drawing, and we can end up with something like this, or at least Isha did. Or if we start with some molten metal, we can turn it into a sculpture, like this one by Chong Fan Chong, that you can see on the Singapore riverbank next to the Fullerton Hotel. I'm sure you're familiar with it. It's easy for us to see where the creative process has come in this, turning something into something much more appealing. So where then do scientific endeavour and creativity collide? Well, I hope to show you that they're in fact intertwined. Think about it, every day you use a smartphone, you drive, or your parents drive, a fuel-efficient car, or you see people, or you take drugs to treat an ailment that you're suffering from. These have all been created in some way by scientists in a creative step to invent or develop them. Scientists must have a solid understanding, right? That's where we start. We need to observe. That means we read literature, we read books, we attend seminars and conferences, we learn about what the latest knowledge is. And then we've got to either pick holes in it or develop questions to take it to the next step. So critically thinking about it and creatively thinking about what the questions remaining are is an important step. And an example here is my PhD supervisor, Professor Nick Reed from the University of Edinburgh at the time, once told me that science will never be finished. And by doing science, you do a lot of experiments, and you may well go a long way to answering a specific set of questions. But in doing so, you will ask another 10 questions. The job will never be done. So based on the knowledge that we have in the world, we then formulate a hypothesis. This, I argue, is a creative step, thinking of a new idea or a new explanation for a phenomenon. We then have to design our experiments, and again, this is a creative step. We have to think of new ways to approach an experiment or a question to address it in a different way. And if we just did the same experiment again and again, we'd just keep finding out the same answers to the question again and again. There are many tools and techniques that we have available to us as a scientist, and selection of the right ones, understanding their pros and cons, their advantages and their limitations is a key step into designing a good experiment. And the tools themselves have come from somewhere. They've been invented and developed, and that is an area of science that involves creativity. And I'm going to draw on examples from microscopy, an area of my own expertise, of where invention has been required and how it continues to this present day. A gentleman called Anthony van Leeuwenhoek is widely credited with the invention of the first microscope. Recently, he would have celebrated his 384th birthday, should he still be with us, as Google celebrated for us. So that's him on the right-hand side. And what he did was look down his microscope, which is what he's holding, and look into a droplet of water from a lake that he got from nearby. And he saw what he described as little animals. These little animals we now know are bacteria, fungi, and algae, or the microbial world around us. This is an image of one of Van Leeuwenhoek's original microscopes, and this is a far cry from the sort of microscopes that I'm using today, just across the road in the Institute of Medical Biology. And this brought to mind a very nice quotation from Oren Harari, who's a late professor from the University of San Francisco, when he said, "The electric light did not come from the continuous development of candles." It's a big step, right, from this to where we are now. And that's an important statement. We make many incremental gains, but sometimes it's a jump into the unknown or a bigger jump forward that allows us to have access to the sort of tools that we use in science. The example I'd like to give you of this is super resolution microscopy. It's a technique that was only developed in the last decade, but has revolutionized some of the biology that we can study as a consequence of its development. And such was its impact that these three gentlemen were awarded the Nobel Prize in 2014 for that development of super resolution microscopy. Without this level of creativity, curiosity, and invention of these scientific tools, we'd still be sat there wondering what all those little animals are inside the droplets of pond water. So here's an example of how we use super resolution microscopy. What you're looking at is a cell that's undergoing the critical step of meiosis, a cell division that leads ultimately to the production of the sperm and the egg cells in mammals. And you're looking at two proteins that have been labeled in green and red. And the red one is called SCP3, and the green one is called Cache5, just the details. What's happening is that the cell has to align its pairs of chromosomes together and then deliver those chromosomes into appropriate daughter cells. This is a critical step. If it goes wrong, it can lead to infertility or genetic disorders such as having a wrong number of chromosomes, trisomies, like Down syndrome. Very important step, and it must get it right every time. So what you're seeing here is a protein, SCP3, which is the glue between the two chromosomes. The green protein is responsible for bringing those chromosomes together in 3D space and holding them together using the red protein. This is a very nice image, but it's a conventional microscopy image before super resolution. What I'm going to do is press a button, it's going to turn it into the super resolution image. So here we go, ready, 1, 2, 3. There you go, exactly the same sample. And what you should have seen is that each of those single red filaments has become two parallel filaments running together that we just couldn't see before we had super resolution microscopy. We knew about it from other techniques, but we couldn't visualize it in such a way. Obviously now we have access to these tools. We can ask the question, have my chromosomes paired? Have they not paired? What's gone wrong? All these sort of questions that we just couldn't access before. So the invention of this technology, the creation of this technology is a very important step for us. It's enabled us to do this sort of work. But thinking more broadly, to get to this stage, we've had to have many inventions along the way that have allowed someone then to develop the super resolution microscope. And they weren't ever thought about in terms of developing them for this application. They were invented for themselves, and then people have thought about how to utilize them. So we needed the laser developed by Theodore Maiman in 1960. We needed someone to develop ways of producing very specific antibodies to label our proteins. We then needed fluorescent molecules to allow us to see them in a microscope. And we then we then needed a powerful computer technology to allow us to drive these instruments. And within those computers, we needed algorithms first developed for the Hubble Space Telescope that we can now use inside a microscope. And finally, we needed those Nobel laureates to think about clever ways to integrate all of this knowledge and put it inside a super-resolution microscope. And then finally, very importantly, and that's last but not least, we needed a creative scientist. And in this case, this gentleman's name is Henning Horn, and he was the lead author on the work that I just showed you. We needed a creative scientist who had the ingenuity and invention to think about using these tools in this novel application. So at every stage in this, scientists have been creative. One further example I'd like to give is that of GFP, very cleverly named green fluorescent protein, because it's a protein that's green when it fluoresces. It was invented not by humans, it was invented by this jellyfish called Aquaria victoria 160 million years ago. For the rest of that 160 million years, the human race as Homo sapiens have had to evolve, and we've had to form a civilized society that does science and other things. And then we found out about this protein that the jellyfish produces. In 1994, Martin Chelfie, the gentleman in the middle there, and his colleagues published the first paper that utilized GFP in the journal Science, as you can see here. Now, the secret to this is that the jellyfish encodes a protein called GFP from a gene. If we can take that genetic sequence out of the jellyfish and put it somewhere else, in this case in a worm, and we can tag it to a gene of interest in that worm, so that the two go together. Whenever it produces this protein, it also produces a GFP, and the two go together. So not only can we now see where our protein is inside the worm, but we can also see where it goes over time inside a living organism. This was revolutionary for biology, for cell biology particularly. We can now label any protein of interest that we want and follow where it goes over time in a living cell. And for this, these three gentlemen here were awarded the Nobel Prize in 2008 for that discovery that has far-reaching consequences within biology. I'll give you two examples of how we use GFP in the lab and its derivatives. On the left here are some human skin cells that we're growing in a dish, and we're watching them behave on the microscope. We've labeled the keratin filaments within those cells. Keratin is not only in your nails, it's the stuff that gives you the mechanical strength in your skin. If that goes wrong, there's all sorts of terrible diseases as a consequence. So now we can watch, as a cell migrates, how that keratin remodels and how it reorganizes itself during cell division and cell migration. On the right-hand side, you have a zebrafish embryo. So this is a fertilized embryo that's going from that single cell and all developing very rapidly on its way to becoming an adult fish. And what you can see here, we've labeled with GFP a protein that's within the nucleus. So you can see the nucleus as the chromosomes condense and then divide into two, and you see the cell pinch off into two daughter cells afterwards. And without GFP, we just could not see this. So coming back to the scientific method, creativity is a key step all throughout this method. Formulating a hypothesis, developing tools, and then choosing which of those tools to use and how, and coming up with an experimental method, and applying them in novel ways. And then it's also important in developing a conclusion. Now, this shouldn't be taken too far. We don't want to create results. We want to use creativity to understand them and come up with ideas of how the world might be working around it. So the work that I can do and others can do is the culmination of many years of research across many disciplines, all the disciplines, by many talented people from all over the world. Each scientist is building on what's known and using their insight and creativity to push forward the boundaries of human understanding. With that, I would like to thank you for your attention and enjoy the rest of today's